So hi everyone. Sorry, it seemed I wasn't I was muted. So as I was mentioning, today's session for anyone who is joining the call will be on the SmartBulk app. We'll try to use sort of our SmartBulk sort of application to be able to identify sort of the efficacy of stem cell transplantation in Crohn's disease patients using sort of our bulk RNA pipelines in the system. So as I was mentioning at the sort of towards the end of the session, we will sort of ask any users if they have any questions. So you can add all of those questions in the Q&A chat, and either I or my team will be able to respond. We'll also be sort of circulating a survey to get some of your feedback on today's session, as well as what you would like to sort of see and your specific case study biological stories you would like us to show as well. So we'll be sending that sort of circulating it towards the end. Okay. So as I mentioned, today's session will mainly focus on sort of Crohn's disease and specifically looking at hematopoietic stem cell transplantation as a form of treatment for these patients. So for any users in the team that are not really sort of working in the inflammatory disease landscape, I'll give some overview of what Crohn's disease is and how this fits into the narrative for today. So Crohn's disease is characterized by chronic inflammation of the gastrointestinal tract. Often this leads to progressive bowel damage and then disability in these patients. This multifactorial disease involves sort of the dysregulation of the innate immune system, which is triggered by environmental factors, which genetically are susceptible in these individuals. So some of the key pathogenic mechanisms include impaired intestinal epithelial, burial, pro-inflammatory, cytokine production, sort of impairment, and as well reduced microbial diversity. Current treatments do include sort of analogous hematopoietic stem cell transplantation, which will be the focus of today, sort of analyzing further the efficacy of this treatment. And basically, this type of treatment stems from the patient's stem cells, which are collected from the patient and then reintroduced to develop a new sort of immune system defense for the patient. So this will be sort of the focus of today. And I just wanted to give an overview of sort of the treatment so you get an idea when I'm going through the session, what we'll try to be analyzing. Okay, so a bit more on sort of the data that we have currently for the study that we'll be using in this case study. So today's session will mainly be analyzing a bulk RNA-seq data set, which includes 36 normal healthy donor participants, as well as 44 Crohn's disease patient samples. These sort of 44 patients underwent the HSCT, so the hematopoietic stem cell transplantation, and we'll try to examine both the effect of this sort of stem cell transplantation in the immune system, particularly in the CD patients, since these are the ones treated. And we'll try to investigate whether these treatments sort of have an effect and how do these effects sort of last. So is there a time point specific sort of effect that we are able to sort of analyze using the SmartBulk app, and we'll try to see if it lasts up to 2 year post-treatment. So this will mainly be the data that we'll be using for today's case study. So again, some of the recap for anyone who is joining. So we'll be mainly sort of concentrating on the differences in the peripheral tissue immune cell populations following this stem cell transplantation, and we'll mainly be focusing on Crohn's disease patients only. So for this session, we'll be subsetting all of the healthy donor data since it's not sort of a specific research aim that we're trying to sort of answer. Okay, so a bit more on sort of before we go into sort of the bulk SmartBulk app, I do want to recap for anyone who's really new to Bioterrain and is not sure like what the platform is, how to navigate it to sort of then perform the type of analysis we're going to show today. So the ecosystem for Bioterrain, it sort of encompasses a multi-level omic sort of research platform. So all of these platforms here have a very specific research need. So whether we're supporting scientists in analyzing their own proprietary large-scale single-cell RNA-seq data sets without any coding backgrounds. So this could be suited for wet lab scientists who really just need support in running normal sort of functionalities. So they can use the bBrowserX platform for that. If there's users or scientists in the team that are joining that do not or are not generating any sort of single cell RNA-seq data or imaging spatial transcriptomics data, we do have our Talk to Data platform, which does encompass all of our sort of large repository of curated cross-study data. So this includes different single cell RNA-seq assays, as well as different spatial transcriptomics technologies, ranging from bulk to single cell resolution spatial and proteomic spatial. So all of the studies can be found in Talk to Data based on the species that you're working on. So we do curate human, mouse, and non-human primate data in the system, which you can then use to sort of create atlases or get insights from the system as well for your own analysis. The Lens platform would be if there's any scientists that are generating spatial data, they can use the Lens platform for uploading and submitting that internal data and then running further downstream analysis from there. We do have other needs. So the session for today will focus mainly on the SmartBulk app, which I did mention today, looking at a very specific case study in Crohn's disease. But if there's any scientists that wanted to know if we have any bioinformatic sort of needs, we do have BioStudio, which does help run AI stacks. It helps run specific ready-to-use notebooks that might integrate different AI modeling or different machine learning algorithms that would require sort of the coding background to run those on the data. So this would be sort of another platform we also have, and we'll probably be using it as the next sort of case study in our sessions. Okay. So if I go into sort of what we're going to concentrate today, we will be going through specific research questions. So the first part of the session, which will try to look at using ANOVA differential gene expression in the SmartBulk application, we'll use these specific sort of features to both examine the effect of the HSCT treatment on the immune system of the CD patients, so Crohn's disease patients. And the second part of the aim, which would be looking more towards the differential gene expression analysis, as well as a heat map capabilities. This will give us insights into our second question, which is investigate whether the treatment effects last up to 2 years post-treatment. So we are going to want to see sort of differences in the treatment stages of sort of this specific Crohn's disease sort of implementation. Okay, so I'll go into my next sort of tab here, which is sort of our ecosystem landing page. So if we want to go into sort of the SmartBulk app, you can directly navigate that specific sort of platform. So you can explore the SmartBulk app, and you can see that it directs you directly into the interface of SmartBulk. So how can you navigate this? You'll have to sort of select what species you're mainly working on. So whether you're loading human or mouse samples, and how you're working in terms of submitting the data into the system. So whether it's on your personal workspace or on a group research workspace as well, so that you can share the data. So I'll show you a bit more how you can submit the data, just in case there's anyone in the team that is new to SmartBulk. So when you're creating a new project, basically the data formats that the system will request or will need is the expression matrix and the metadata file. So we do convert the gene symbols as well from the RNA bulk data, but you'll need to make sure that these two files are in the correct format in order to upload them into the system. So the first thing that you'll need to make sure is that you're adding sort of a name to the data set that you're going to load into the system, that you're loading it into the correct species database as well, and how you're sort of sharing the data. Okay. From there, we can go towards sort of selecting what file to upload. So the first file is the matrix file that I have here. So it's a CSV format, but we do support other different types of formats, for example, TSV or text files as well. So you can see sort of more information on the extensions. But as I mentioned, the first step will be to open up the data set and load that data set into the system. Okay. So I might just start with the metadata first. Okay, that might just be an error on my end. But when you submit the data, you should be able to see sort of all of the different pipelines being run. And since this does take a few minutes, I did want to sort of use the time correctly. So I do have a already preloaded submitted data set, which is the same data set that I was trying to upload now. So just make sure when you are loading that it's correctly in the file format that we need them. So if I go back to SmartBulk where I should see the submissions that are being performed, I can then explore that specific study in the database. Okay, so the first thing that you'll see when you upload the data is sort of the input and quality control tab, which would give you sort of the information relating to the original data that you've loaded in the system. So this would be the first thing that you're sort of seeing in the system. So both the metadata and sort of information relating to the quality control. So what can we do from here? In order to make sure that the data set has passed some QC metrics and that you're making sure that you're subsetting low quality samples or subsetting specific labels that you need for the downstream analysis, you should be able to then perform the quality control on there. The metadata would just give you an overview, just to sort of verify that you have all of the sort of labels that you need for your analysis. So when the study sort of or the data set is loaded and submitted, you should be able to then confirm that all that information has been loaded correctly into the system. So you can then sort of move forward. So from here, the next step is doing the quality control. So I want to be able to verify that there's no low quality samples based on the sort of RNA transcript information. So a way to do that is being able to add your own matrix filter QC parameters. Some of the parameters I'll be using today are sort of gold standard for this specific sort of tissue and this data format that we're loading today. But in terms of what you should add, it should be specific to your data, the type of format that you have the data in, and the specific case of the data. So for here, I want to be able to find or keep the genes expressed in at least 5 of my samples. So I want to make sure that sort of input is included. I want to also be able to see that these genes are within at least 10,000 of my samples. So this would be sort of the gene that I want to sort of have an input in. And then the normalization methods, this is really up to the user if they want to apply any further normalization, or they might already have data that has been normalized. So this is really up to the case of the data that you're loading into the system. But for my case, I want to run log 10 normalization. I don't really want to remove any batch effects. I don't really need that at this point. But I want to subset the data as well. So as I mentioned, I'm not really concentrating on healthy donor samples. I'm mainly focusing on Crohn's disease. So if I want to be able to subset that data, I have to make sure that I'm only including the Crohn's disease samples for this analysis now. Okay, so once all of this input has been sort of updated, I can then update the plot as well. So this is just going to take a few seconds to run in the pipeline. Okay, so I can see here the PSA sort of corrected QC samples, and I can see that there are some samples here that are drastically shown as outliers. So what can I do from here in terms of how can I correct the data further? So to be able to sort of investigate this PSA, so what are these PSAs showing? This is basically visualizing the data variance and also the outlier samples, which points to what samples are in the axis that are capturing the major sort of variance sources. So the PSA plots assess sort of are clustered by condition. So I removed normal, and I can see sort of more information relating to the quality control that I can then use to sort of either remove low quality samples or then sort of run the analysis from there. So the way to sort of get more information as to like what samples have this low quality, you can go into the count blocks plot and you can directly see the information relating to the raw count and the normalized count. So I can see here that there are some samples that are not meeting sort of the variance, similar to the other samples that have the same similar variance that I want to see. So for this sort of outlier, if I sort of go to this sample, I can directly remove it from the data set so that it's subsetted with the sort of high quality samples that I want in the further analysis. So you can see even from going from the normalized count, it was able to sort of normalize the data so that you have a higher quality as compared to just using the raw count data. So that's another reason why we recommend to perform some sort of normalization method on the data. Okay, so once we sort of excluded samples that we don't need, or we subsetted the data that we are going to use, we can then sort of conclude the subsetting of that data set. So this is sort of a crucial step because it is going to give you then the new information with the samples that you actually need that have been sort of passed through the quality control step. Okay, and this sort of ensures that you have sort of the reliable samples for the downstream analysis. Okay, so as I go from here, the first step that we'll try to perform is the ANOVA analysis. So as I mentioned, if I go back to my slides, we were trying to examine the effect of the HCT treatment on the immune system of the CD patient. So this is the first question that we want to answer. So we want to be able to see if there are any sort of differences in response to non-response for this treatment. So if I go back to the data, I'm going to go to this ANOVA function, which is directly sort of access to the smart bulk. And I can add more information as to what I'm trying to analyze here. So here I can do CD patients, subset response to non-response. So this would be my title. I can then select what data set I want to include as well. So this, for example, if I'm selecting the normalized data, for example, this one that I just performed, one thing that you should keep in mind is that in terms of sort of the statistical methods, which we'll cover later on, there will be specific methods that are only available for normalized data. So we'll be covering a bit more information on that. And the basic column that I want to include is the data relating to the response to treatment. So I want to be able to identify if there's any differences in the HSCT response from these CD patients. All of these parameters are default, so you should be able to see this as well on your end. But we will be adding other features depending on the type of data that you're working on. So when we submit this analysis in the system, and I'll just add this information here, this will take a few seconds to load. So I do have here already prepared ANOVA result to just save time. So once the ANOVA result has been returned, this is the information that you'll see. So you'll be able to see sort of the information relating to the log p-value of these genes across the different chromosome numbers in the data. And basically the first step that you want to do is be able to identify if those marker genes are significant in this data. So I'm going to add an adjusted p-value of 0.05. And I'm going to want to see if I'm able to identify any specific inflammatory driven genes from this specific response to non-response comparison. So from here, since I'm mainly looking at the chromosome count as well, I'll sort of change the chromosome. I'll adjust the p-value so I can sort of identify these highly significant genes. And I can see here that there are very specific inflammatory genes that I sort of recognize based on other Crohn's disease studies. So for example, the HLA-DQ2 is quite a known gene to be sort of differentially expressed or deregulated in these samples. So I can definitely see that there is a very clear difference in the response to non-response group, which is significant. So you would be able to map these as well. If I go into other ones, so STAT1 is another good example of sort of an inflammatory driven gene, and I am seeing some downregulation again in the response group. And if we go further down the line, another known sort of gene is CD40 as well. So I can then sort of map these based on the chromosome number and based on my p-value adjusted number as well that I've added here. So what does this tell us? So this observed sort of reduced expression in responders indicates sort of a lower inflammatory response in these patients. So why is this sort of important in the context of just sort of the hematopoietic stem cell transplantation in these patients is that there is sort of a downregulation of these inflammatory genes, which suggests that there's an effective modulation of the immune system post the sort of HSCT treated patients. So this sort of aligns what we sort of observe as a reduction in this inflammatory expression, which also sort of may lead to some sustained remission. And then that would sort of further positive clinical outcomes in sort of these patients that are developing this chronic inflammation disease. Okay. So if I go back, so we got these results from the sort of ANOVA function. If I go back, so okay, my second sort of question from here is to investigate whether the treatment effects last up to 2 years post-treatment. So I want to be able to then analyze from the responders group if there's any differences in the time point of the treatment. So if I go back into sort of the analysis, for that sort of question, you'll need to perform the differential gene expression analysis. But before we go there, we need to make sure that we're going to subset only the samples relating to responders. So I'm only interested in sort of the response groups. So if I go back into the metadata that we created, so the QC sort of applied metadata, we can then create a new subset from there. So I'm only interested in sort of Crohn's disease, but in this case, I only want the response samples so that I can then look at the differences in the sort of type point and see if there's any sort of relation to post 2 year treatment. So from here, the first step would be to then perform the quality control again. So I'm not going to sort of apply any further normalization steps or any other filtering because we've already applied this in the first stage of the analysis, but I will want to subset the responders from this specific data set here. So I have one condition, and then I'm only interested in the response group samples. So I'll update the PCA plot so that I'm able to then subset the data. Okay. Okay. So once this has been completed, I can then subset the response samples in the analysis. So this will take just a few seconds to run. Okay, so now I have this sort of subset. So I can confirm that I have my Crohn's disease and that I only have my response to treatment samples. So from here, I then want to sort of answer this question by performing the differential gene expression analysis. Okay, so the first step that you'll have to do is make sure that you're selecting the correct sort of data set. So I want to select only the last driven data set, so the one that's 219 that has been subsetted from the response treatments. But why can I not see my data set here? So I can't really select one. So the reason for that is that for DSeq2 and EdgeR statistical methods that are used for differential expression analysis, normally these type of methods are designed for raw count matrix data. So they're not sort of suited for normalized data. Since I did run the normalized log 10, that's the reason why I can't select those statistical methods. So that's just something to keep in mind, that these specific tools rely or assume that the certain data has been distributed a certain way, which typically is a negative binomial distribution. So for only raw count matrix data that you might want to sort of include in the system, you can run the DSeq2 and the EdgeR. Since I don't have the raw count and I did perform QC to remove any low quality samples and also to subset the data, I can only sort of run it on Lima, which is the other statistical method that is suited for this normalized count data. So I can then select my subsetted data that I've run in the last session, and I'll perform the Lima statistical method for that. The next step is to also include a primary factor and a reference factor. So the primary factor in this case will be in relation to the week of that time point. So in relation to week of the hematopoietic stem cell transplantation treatment. And then the reference factor will be to my baseline. So I want to be able to compare all the different treatments to my baseline time point 0. And there's no other batch information I need to add from here. The only thing that I should add is what am I running the reference factor to? So what is my time point 0 going to run towards? So it's going to be run for the other time points in relation to the treatment post the HSC2 in these patients. So once I have this information here, I can then submit the analysis, and it shouldn't take too long to run the analysis. So again, it takes around 5 to 10 seconds, depending on sort of the scale of the data that you're running. Okay. So once this is run, the first thing that you'll be able to sort of visualize from the data is a summary table showing all of the differentially expressed genes in this case. And you'll also get more information as to what genes are downregulated. So one thing that I wanted to gather, okay, I do see that there's a downregulation in these responder patients. So I got that from the ANOVA result. And I want to see if there's any indication where it's based on a specific time point where I'm seeing this downregulation. So once the differential expression has been run, I can then sort of see all of my list of differentially expressed genes here. I don't need to add the adjusted FDR, so it already has the 0.05, but I can then sort of map based on sort of my genes of interest. So some genes that I have seen from previous sort of information relating to hematopoietic stem cell transplantation is P13. So that's a well-known gene as well that is showing, and we can see that it is sort of significantly downregulated. So if I start with the first time point, so let's say I want to be able to see if we're able to see this downregulation at the start, so week 26, as compared to baseline time point zero. So I am able to see this significant sort of score. So if I then want to match it with other sort of inflammatory related genes, so I can look for another gene, sort of S100. So that's quite a known gene as well. So you can see here all of the genes, and then I'm mainly interested in the one for 8. So I can include this gene as well. Another gene that has also an inflammatory sort of role is IL-1B. So that's been quite known in some Crohn's disease sort of data sets as well. So now that I have sort of these genes here, I can see that these genes have a downregulation from week 26 as compared to time point zero. So if I then want to be able to change into week 52, so I want to sort of analyze if this downregulation is maintained based on these inflammatory markers, I can then go into week 52. So what am I seeing here? So I can definitely see that this sort of downregulation has been sustained even going into week 52 post-treatment. And this is significant based on the genes that we've queried in the system. So I can still see a relation with the MP13 sort of gene. I can also see it in relation to the S100 gene. So I'm able to also see this in the plot as well. And there's a lot of genes that have been shown in sort of week 26 that are showing up as well in week 52. So this is showing us that this sort of sustained suppression does suggest a benefit from the hematopoietic stem cell transplantation that extends from the initial stage post-treatment period. So from there, if I want to see whether this leads into long-term post-treatment, so we want to see based on the week 106, which is the last sort of treatment stage, and I want to see if this long-term remission and reduced need gives us some information relating to the intervention. So if I go into week 106, what am I sort of visualizing here? So I can see that there's still sort of a sustain. So it's clear if I go down sort of the list, I can query the genes that we were looking at previously. So the P13 gene, and I want to see if it also sort of is in the list, or I want to see if the S100, A8 has a sustained sort of role. So I can still see even from week 106, and let's say if I add another gene just to make sure that I'm getting the same analysis, so the IL-1B gene, okay, I can still see that there's also a sustained sort of downregulation. So what is this showing us even from week 106? So we can see that the downregulation of these inflammatory genes are preserved, even going towards the last stage post-treatment, which gives us the indication that the long-term dysregulation of these key inflammatory genes serve as sort of mediators in and sort of has a durability in the HSC2 effects, which can give a role into the long-term diseases and how can we manage these patients and their quality of life based on sort of this information. So this is sort of the insights that we can gather. And what else can we gather just from differentially expressed genes? So if we then want to see if there's any indications relating to other cytokine inflammatory genes, and we want to map that in the heat map, we can go to the heat map result and we can either map based on all of the dysregulated or downregulated genes, or we can bring up our own sort of cytokine markers as well. So some of the known markers that have been known for like Crohn's disease or just stem cell transplantation modulation are these that I have here in my list. So TNF, IL-A, IL-B, IL-6. So these are sort of core inflammatory markers, and I want to see if there's any indication of sort of their inflammatory role and their downregulation in week 106. So I'll update the heat map so I can see it in the clustered heat map results and what other information I can gather from here. So what can I see from this? So we can see just from my own sort of gene cytokine marker list that up to 106, 106 weeks post-treatment, we can still ensure the efficacy of this stem cell transplantation. So why is this important? So the persistent suppression of the cytokines like IFN NG or IL-17A or IL-6, for example, is an indication of the reduction of the systemic inflammation. So we can directly see this being shown in the heat map, which is critical for maintaining remission in these patients and sort of stalling the disease progression as well. So these are sort of insights you can gather. And again, you can look at the different data. You can input your own gene sets as well, or also select if you want to adjust the p-value in this data set as well. Okay. So if I go back to how can we take this analysis even further? So if I go back to the slides and I want to see other questions that I want to identify. So from here, I want to identify if there's any commonly regulated genes across the different time points that I can then target or sort of use for immune modulation purposes. And the fourth sort of session would be to uncover if there's any biological pathways that have a functional role that are associated to this treatment response. So based on sort of the geo system, if we can gather any information relating to this pathways. Okay, so if I go back into the analysis here, I can then run this type of analysis using the Venn analysis diagram. So from this sort of this data set here, I want to be able to analyze the co-regulated genes. I can add my analysis, so co-regulation, and then my set. So what am I going to run the analysis from? So I'm going to run them based on each different time point. So week 26, week 52, and week 106 to be able to identify these genes. And then I can submit the analysis for the Venn diagram. So just to save some time, if I pull up this analysis here, I want to be able to identify specifically if there's any co-regulated, downregulated genes across the different time points for the Venn diagram. So if we go then to the downregulated genes, I can significantly sort of find that there's 190 co-regulated genes across the different time points that can be further used for immune modulation or targeted. So what can we sort of gather more information from here, aside from just the log fold change or just the fact that we identified these 190 genes? We can then run a further enrichment analysis from these genes. So when you go into performing a new enrichment analysis, you can select what database you want to perform it from. So the DGN that I'm sort of typing here is based on just the disease-related processes. So you'll get an idea of what other inflammatory processes are showing a correlation with these genes. And then I can add the GO-all pathways as well. So I can get both analysis for this Venn diagram and I can submit it into the system. So I do have a result here, which will save us some time. So for the DGN, which is mainly suited for the disease-related processes, I can see that there is a direct role from these co-regulated genes across specific inflammatory-related diseases. So I can see that these are mainly downregulated in inflammation. We can see other inflammatory diseases and other sort of diseases related to the lymph node that also has an inflammatory effect. So this is sort of the information you can gather relating to sort of the p-value as well, and sort of the system disease-related factors. If we go into then looking even further into what specific sort of immune system pathways we're able to see in this downregulation, we can see that these 190 co-regulated genes have an effect in very specific sort of pathways. So why is this important? So we can see that there's a downregulation in the genes that are involved in this inflammatory response, which suggests that this type of treatment reduces the chronic inflammation in Crohn's disease and other types of diseases that are sort of targeting the inflammation system. We can also involvement of these genes has a positive regulation, specifically in the cytokine production, which indicates that sort of this treatment might have an initial boost in the immune system as well. So over time, this sort of analysis could be used to then regulate the immune system and sort of prevent this excessive inflammation in these patients. Okay. So if I go back to the slide, I do want to sort of gather some concluding remarks. So what did we find from this sort of analysis using the SmartBulk app? We were able to identify that up to even 2 years post-treatment, there is an inflammatory response that has been stably reduced through the downregulation of very specific inflammatory gene markers. So we were able to find that even just from the ANOVA. We were able to then replicate that in the differential gene expression, which we wanted to see based on different time points. And then we were able to also replicate this when we were trying to look at how these co-regulated genes have an effect or a functional role in the inflammatory pathway system. Okay. So a bit towards the end, if there's anyone that has any specific questions, since this does conclude the webinar today, please add any of your questions below so I can answer those in the Q&A. And also my colleague has sent sort of a survey that you can fill in. So we can gather some insights on what you would like to see for the next sessions or your specific feedback for these webinars. So I'll go into the Q&A just to see what questions we have currently. So beside this sort of attendee had a question, beside the differential gene expression and enrichment, what kind of analysis can be provided from the SmartBulk app? So another sort of based on the roadmap that will be having other functionalities, will be including the pseudobulk deconvolution feature in the SmartBulk app. So that will be a new feature that will be added. Another one will be sort of having the batch effect removal for the Lima package. So that will be another feature that will be updated very soon as well. And another feature that has been asked from previous scientists is the WGCNA, weighted gene co-expression network analysis. So this will also be added in sort of the SmartBulk application pipeline as well. So these would be other features. So another question is, I see that there's a free curation program, and I'm not a Biotrain user currently. Can I request public data to be curated and try the app? So to answer this question, yes. So we are running a huge free curation program for bulk RNA data. And this is across any users, whether you're an existing researcher using the platform or a new user that wants to try the platform. You can contact us at the support at biotrain.com, and we can give you access to the system, to all the platforms in the system that you can try. And you can also sort of request any unlimited number of bulk RNA public data that you want to use for your analysis in the application. So this goes for existing users as well. We are running this program until September 30. So please take advantage and please send us any list of data sets that you want us to curate that you want to use for your analysis. Okay, I see that you provide 3 types of differential gene expression. How should I choose these methods for my data? So this is quite a good question. So what method you should use is really dependent on the case and the type of data you have. So as I mentioned previously, if you have raw rank data, so non-normalized data, for this type of data, you can run the DSeq2 and the EDGE-R. So you would not be able to run normalized data using these methods. So the third sort of functionality that you would is the Lima package, which is quite suited for normalized data. And for any users that have any microarray data, this is quite suited for the DSeq2 or the Lima package as well. So it really depends what application, what assay you're using or technology. And we can give you further details if you have a very specific case. Just reach out to us at support and we can give you exactly what method or what parameters we would recommend for your specific research data that you have. Okay. And is there any QC for the data, also future inclusion of proteomics data? Debbie, that's a great question. So we are sort of trying to add other functionalities relating to proteomic data. I'll add that as a request to our team. And I'm sure we can follow up, especially our lead on the SmartBulk app, on how you can sort of attach QC parameters for proteomic data as well. We'll definitely follow up after the session for your specific question. Is there any recommendation for gene filtering? So yes, there is. And again, that really depends on the type of data as well. So normally the ones that I selected, for example, the gene filtering up to 5 sample, this was mainly because this helps reduce the noise in the sort of lowly expressed genes in the data. So normally a lot of bulk RNA-seq will have like at least in 5 samples. The 10,000 genes is quite a high score. So if you have other sort of gene coverage that you need based on the sequencing that you're performing, again, you can reach out and we can sort of give you a specific filtering recommendation for your data. Okay. So I guess if there's any other questions that weren't be able to sort of answer in this session today, we'll follow up and our team will be able to answer those. And again, please sort of fill out the survey. It just gives us an idea what other stories you would like to see and any feedback in terms of how we sort of run the webinars as well. Okay. So thank you everyone for joining the call. And again, as a reminder, this is a weekly session, so you'll be able to get sort of the reminders of our new story and what we're going to cover for the next session as well. So I hope to see everyone in the next session. And thank you everyone for joining.